Hello, everyone. Thank you for joining this very interesting panel, which is dedicated to precision medicine and which is funded by NM Genomics, one of the companies where I am part of the founders team. In fact, I'm very happy to broadcast from Vienna. This is why I am wearing a traditional Austrian Tracht. It's a classic jacket for people who are well off, and I'm very happy actually to start this meeting right now. We have fantastic speakers once again and very interesting topics. Now it's my pleasure to hand over the moderation of this event to Veronica, one of our senior employees. Veronica is a molecular biologist with huge expertise in lifestyle testing. So Veronica, the floor is yours, please.
Thank you, Dr. Georgiev. Welcome to everyone. I hope you're all feeling all right. It is of utmost pleasure for me to moderate today's discussion, where we will talk about the importance of nutrigenomics and the health of our microbiome. So welcome everyone to the discussion, the microbiome and nutrigenetics. Before we start with our first speaker, I would like to remind everyone that each panelist has 10 minutes of presentation time, and I would kindly ask you to respect this time period so that no one is left behind and we have plenty of time for a nice discussion in the end. So just to make sure everything runs smoothly, I will give you a note when the 10-minute mark is up, all right? Thank you. And as for our participants and attendees who are currently watching, everyone is welcome to leave their comments, their questions in the chat during the talks, and I will do my best to share it with our speakers. Okay, so let's start. Our first presenter today is Dr. Olga Antonova. She is the Chief Professor at the Department of Medical Genetics here in Bulgaria in the Medical University of Sofia, a board member of the European Society of Human Genetics, and associate member of the Public and Professional Policy Committee. Her interests are in the field of disease prevention, precise medicine, and health consequences of interaction between the human genome, nutrition, and environment. Dr. Antonova, the floor is yours.
Thank you, Veronica, for such a nice presentation. So, okay, I maybe should share my screen now. Okay, I hope it's okay now. Okay. So, yes, good morning. My name is Olga Antonova, and today I would like to introduce you one very interesting, I hope, topic, genetics application in nowadays medicine polygenic approach. So 20 years ago, only certain persons suffering from specific rare genetic disorder or those with high risk either to develop or to transmit genetic diseases could take advantage of the genetic technology. Nowadays, genetics benefits in the rest of the society. It allows to reveal the hidden risks in each organism. This risk could be related to social significant disorders like diabetes, mellitus type 2, atherosclerosis, some common cancers, even some food intolerances like gluten intolerance, coffee intolerance, lactose intolerance, as well as the need for additional supplementation of some group of vitamins like vitamin B, vitamin C, D, and etc. The workflow actually is now very easy and comfortable for the end user. We have collection of genetic material that is done non-invasively. You can do it at home. Then this material is should be sent to the lab. We have performing the genetic test itself, and then afterwards we have bioinformatic calculations and determination of the risk category. Afterwards, we have maybe the most important part of our process. This is actually the real counseling process between the certified genetic counselor and the patient or the end user. So at the end, we have explanation of the results, genetic results to the patient. So to be sure that he understands all the genetic risks and power and how to use this precious information. You may be thinking now, okay, it looks very interesting, but is it real? So in the next few slides, I will try to explain this from a scientific point of view. When we study DNA, we actually study the sequence of genes of interest located in our DNA molecule. You know that these sequences are built up of 4 nucleotides: adenine, thymine, guanine, and cytosine. And as a result, if we sequence the total DNA, we are going to have a huge amount of data. It is actually around 3 milliards of nucleotides. So these 3 milliards nucleotides are actually up to 99.9% conservative. So the differences between each of us accounts only to 0.1% from the total genome. But still, this is a huge amount of data. This estimates around 3 million of nucleotides. And actually, this individuality of 0.1% actually results in the different appearance, different talents, but also some genetically determined risks in each specific organism. These risks nowadays are not only these rare genetic Mendelian diseases or chromosomal disorders, but also these are social significant disorders like obesity, diabetes mellitus, osteoporosis, celiac disease. These disorders are also called polygenic diseases. And actually, polygenic diseases are due to combination of specific genetic variants and environmental factors that can either exacerbate or prevent the disease. Different genetic tests determine the SNPs, so these are single nucleotide polymorphisms of interest, and aggregate the effects of many genetic risk variants across the human genome into a single score, polygenic risk score. Such scores have predictive value for multiple common diseases, and there are multiple areas where this has clinical utility, often actually guiding the clinical management. So now, having this nice tool, we can build up a personalized risk model and prescribe personalized preventive diet. So we can determine the specific damaging environmental factors to adhere appropriate physical activity and timely to perform specific to these risks laboratory or functional test. So our aim at the end is this small fluffy girl not to become overweight lady with obesity, insulin resistance, lack of physical activity, and potential fertility and mobility problems. And now I would like to introduce some nice examples of the power of the preventive genetic testing and the possibilities of using it in our daily lives. So the most pleasant example for me is actually the daily coffee consumption and the risk of coffee consumption and the cardiovascular diseases. So you may be known that over the years there have been many studies on the amount of healthy coffee consumption. In some studies, they say almost stop coffee, but other studies say consuming up to 4 cups of coffee a day is absolutely normal. So, but the truth is that different studies have been actually conducted among different populations that have different genetic background. And now it is well known that coffee is metabolized by the enzyme CYP1A2, which is coded by a gene called CYP1A2, which has faster, so which calls enzyme with fast coffee metabolism and enzyme with slow coffee metabolism. So in combination with genetic variants and environmental factor, which is coffee consumption, we can determine the exact amount of coffee that we can drink each day. The other variants in genes that are predisposing to impaired collagen type 1 production, as well as slower calcium absorption, are also associated with elevated risk factor for, are also associated with elevated risks, genetic risks for osteoporosis, not only in women, but also in men older than 50 years of age. So people with a risk variant should consume more calcium and vitamin D, as well as less caffeine, because caffeine and calcium are antagonists. And now we can, having this information in mind, we can calculate the normal healthy daily coffee consumption. It could be 1, 2, 3, or 4 cups coffee per day, dependent on the risk variants and additional environmental protective factors. Okay, so thank you for your attention.
Thank you so much. I'm looking forward to some interesting questions in the end of the discussion.
So
Our second speaker today, it's okay, we'll, our host will mute you, no worries. Our second speaker today is Dr. Milena Georgieva. She is an associate professor of molecular biology at the Bulgarian Academy of Science and holds a master's degree in cell biology and embryology. She is a scientific consultant at the International Inago Institute Academy that aims to support scientists from all around the world with advice on academic development. Some of her professional interests are in the areas of molecular biology and genetics, epigenetics, and modern biomedicine. Dr. Georgieva, you're welcome to start.
Hello, everyone. It's a pleasure to be here with you. And I think that my presentation, which is focused on aging, let me share my presentation now, is on longevity. And I think it's a nice leap from the presentation of Dr. Antonova, because she very well introduced the advantages of genetic technologies, and I'm going to show you how this helps the aging research. So the topic of my presentation is Spotlight on Personalized Longevity, which is a real hot topic nowadays. And if we look back to our history as human beings, we can see that the longest quest in human history is the search for the fountain of youth. And this idea has been formulated back 400 years before Christ by the Greek historian Herodotus. And if you go back to the Greek medical specialists these days, like Hippocrates, we can see that these people over there, ancient people, only by empirical science have been able to show that 1,800 of health and disease pathologies are considered as a result of the age of the organism and the imbalance in the full body fluids, like blood, phlegm, yellow, and black bile. Later on, 300 years later, the Roman philosopher Marcus Cicero formulated his idea or his hypothesis that old age itself is a disease. Just imagine how smart has been these people at this time. And if you follow the graph, you can see that, yes, we were able to extend the lifespan very importantly, but we did not succeed, no matter that we changed the environment, no matter the medical technologies, we did not change the decay start point in the aging body. And if you follow the graphs, you can see that the decay in the body that is associated with the aging process starts at the age of 35, no matter the prolonged lifespan. So it means that something is missing in the big picture of aging, and here should unite and work in very good complemental symbiosis, all the efforts of the scientists in order to see what's the missing point to don't be able to change this decay start point, no matter of the lifespan. Well, if we look at this table with all these long-lived and living organisms, we can see that we did not succeed in making us living until the age of 200 years. So it's around 122, 117, and so on and so on. And all these people, as a matter of fact, that already have died, no matter that they have been centenarians, they died by age-associated diseases, which means that, okay, the prolonged lifespan is something good, but what's important is the health span, or how these people live these years later on in their individual life as happy and healthy individuals. Well, yes, the maximum lifespan appears to be unchanged yet. So you agree with me that we spend billions of money, billions of euro, dollars and whatever to treat the downstream events of aging. Only in USA this year they will spend more than $500 billion on pensions and $300 billion on treating neurodegenerative diseases and almost the same amount of money to treat cancer. So we spend a lot of money treating age-associated, age-dependent diseases. And we know that only 1 year, calculations has shown that only 1 year of extra life brings huge financial benefits to the whole world population. And yet the fountain of youth is undiscovered, but today is pursued more than ever, because anti-aging research has been a modern field of recent biology and molecular biology and genetics as well. Anti-aging turned into a big business, and there are a number of many startups and companies that pursue one and the same goal. And this is to stop, eliminate, or simply slow down the process of aging in our bodies. And they include the recently founded Autoslobs. You all know the news that was spread all around the world, because these people over there will try to reprogram the human body in order to stop aging and age-associated diseases. And it is very much founded in what we have done during the genetic revolution that has been described by Dr. Olga Antonova. And all down they go to Nikolaus that has been settled 10 years ago, and yet they did not found the fountain of youth. So we need a multidisciplinary approach, or transdisciplinary approach to tackle this. What is very, very important for us scientists and medical specialists is that in June 2018, the World Health Organization released the 11th edition of its International Classification of Diseases. And guess what? The old age appeared as a disease code, which was a very good point in our research. And this boosted a lot of money in this kind of research. And in 2020, the United Nations General Assembly proclaimed the beginning of the so-called decade of healthy aging. So a decade of healthy health span and prolonged lifespan as well. So I believe, as part of this research and following all the trends, that we are entering a revolution and a pivotal period, just the same as we were entering the genomic revolution at the beginning of this millennium. And yes, we have 7 signs, even more, of longevity revolution, and they involve all the efforts that are put toward the personalized medicine and its definition and the way it works on the patients. Also involving the global tech giants that entered into life sciences and made life sciences very important and very, very well recognized subjects for research. Also classifying aging as a disease 3 years ago, and all the interventions that were aimed at augmenting the humans and extending the health span. And yes, the economics of aging research is growing rapidly up. So what is aging from the biological point of view? Okay, we discussed aging. We know that we grow old and this is inevitable, as a matter of fact, but what it is indeed, what's happening, what's going on? It's not only the white hairs, it's not only the physical disabilities. There are many things that should be spotlighted as biological molecular mechanisms that happen in our bodies. Yes, from the biological point of view, the aging is something inevitable. It's a time-dependent decline in all functions in our body. It also involves a series of processes that incurs direct damage to our body, and not only to our body, also errors in the biomolecules are accumulated that we cannot repair, and it's also some of the part of the aging theories. And also it is a very complex process that goes on all levels that you can imagine, starting from the molecular, cellular, up there to the aging organism. And yes, the graph down shows that, yeah, we can prolong lifespan, but the most important aim for us scientists and medical specialists is to prolong the health span, this time when we live our old years happy, healthy. So this is the decade of healthy or happy aging. If you go to the modern theories of aging, because if you want to tackle a disease like the aging, we should know all the theories for that try to explain why this process happens in our body. And we go back to the programmed aging theories, which are very famous. Okay, this is a programmed longevity. We have different theories for aging of our systems. We have the damage and error theories, a lot of wear and tear theory, the free radicals theory, and so on and so on. But what is very, very much famous today and that unites all the others theories is the information theory of aging, which shows that aging is a result of the deterioration of changing the differentiated cell's epigenome. So this is changing the information, all the signals that govern the cell to use its genetic information in a particular level. So this happens on this level and unites all the other theories. So this theory, the information theory, looks at the aging body as a whole ecosystem that starts from the most important biomolecules in the human body, DNA, RNA, and proteins, and go up there to the aging organisms, going through the cells, organs, tissues, and so on, to get this not so good-looking picture of an old man in a wheelchair. So how to make a longer lifespan, a longer health span? So this is one of the very important ideas that we try to tackle or try to make with our efforts. Is just to extend this position of decay in the human body, this position of accumulating this information errors in the way the genetic code is read in our cells in order to have a prolonged health span, which can coincide even with our lifespan. How can we make this personalized longevity? This is a really very, very hot topic at the moment, and it's something that is going at a fast speed. And I always say that the moment I blink my eyes, something very big happens in this field, fortunately and unfortunately, because we cannot catch up. So the personalized approach that analyzes our biological data to optimize our health, improve our lifestyle, and extend our health span is called personalized longevity. And it is personalized because it is focused on all the levels of a certain human body as a whole ecosystem. How can we do this? Of course, by identifying the key drivers of aging. So we should know very well the process of aging. And believe me, this is very well known, up to date, from a scientific fundamental point of view, to analyze the biological age rates, to have the so-called clocks for analyzing these rates at which we biologically age, because there is a difference between the biological and the chronological age, and our idea is to make them coincide, to have a very good biological age at a very late chronological one. So this is the main aim for us. And also to close monitor all the hallmarks of aging and to get this health and longevity active advising, as we already heard in the first presentation. So this is real, this is happening, and there is a very big field for development of this market, of this science, of this technology as well. So we have an urgent need for accurate aging biomarkers, because to tackle something, we need to know it very well. You cannot control something that you don't know well. So a lot of investments of efforts and money are in identifying the accurate aging biomarkers at the genetic, epigenetic, and metabolic level. Also, a lot of precise clocks for biological age are being developed and are developing at this point, and they unite different technology in a multidisciplinary approach, like artificial intelligence, data analytics of our body, biological biomarkers, and so on and so on. And we have in 2013 the biological DNA methylation clock of Dr. Horvath, which has been upgraded today with this artificial intelligence to the deep MNH DNA methylation aging clock, which is based on a neural network, and it's really very, very precisely measures the biological age of a person with an error of 2.7 years, which is anything because we refer on biochemical parameters. And also we need accelerated clinical trials with geroprotectors, the so-called senolytics, with a very big hot topic today. There are so many on the list, you cannot believe in the databases, there's so many senolytics that are used in clinical trials in order to tackle all the genetic, epigenetic, and metabolic aging hallmarks that happen in an aging body. So we have a lot of work to do, luckily, because we have something very meaningful to do. So which are the most common cellular hallmarks of aging? This is an amazing review by Perez-Utin and his colleagues published in 2013 that is cited, I cannot imagine how many times, because it's a very good illustration of what's going on in the aging organism at all the levels of its cells, molecules, and organismal level. So we have a primary hallmarks of aging, which are the fundament of everything that happens in our cells, and it is very much grounded by the error theory, because we have genomic instability in aging cells, we have telomere attrition, we all know this, and we have change in the proteostasis, and also change in the information theory, which is called epigenetic alterations here. We also have compensatory hallmarks, which are very important, 3 of them. They are a result of the primary ones, and then we have the integrative that go up to a higher level, upper level of the human organism, and also show a very big decline in aging organisms. So now we know all these 9 hallmarks. As a matter of fact, at the beginning of this year, a 10th hallmark has been promoted also, and this is the stiffness of extracellular matrix, which is a very important phenotype of aging cells, and not only, but it is also very important that people are working a lot in order to elucidate all the fine molecular mechanisms through which the aging happens at all levels that we have discussed, molecular, cellular, and organismal as well. And of course, all these interventions that happen now in the anti-aging industry and research is to tackle all these 9 or 10 hallmarks of aging by certain high-tech drugs and also approaches in order to prolong the health span of an organism. And a lot of clinical trials are underway now, and a lot of them are very much successful because they show some kind of an improvement of the biological age at a molecular level in the individuals that are under these clinical trials. So yes, can we hack the code for immortality? Well, maybe. We did not yet. We extended the lifespan. We succeeded to extend the health span to some extent, but we have a lot to do. And this should be done by the smart touch on big amount of health and molecular data that should be analyzed by sophisticated analytics, neural networks, and artificial intelligence in order with scientists to be able to promote, to translate to the patients, revolutionizing diagnostics, treatment, monitoring, and drug discovery in order to extend our health span. Because the vanity of living forever is something that I believe all my colleagues did not like that much, but what we really pursue is extended health span and also a very good biological age at the later stages of our chronological. And with this, I thank you for your attention, and I'll stay for the discussions after that. Thank you.
Thank you. It was a really interesting talk, and I thank you for making the distinction between lifespan and health span, because I feel that this is a topic that a lot of people don't quite understand. So thank you. I'm sure it's going to be an interesting discussion in the end. Okay, so we move on to our next presenter. This is Professor Sarper Diler, well known for his work in many international scientific and patient groups and committees, such as the European Cancer Organization Patient Advisory Committee and the European Cancer Concord. Dr. Diler has also acted as the president of the MyBioma Patients Europe for 4 years, and as a secretary and board of directors of the lymphoma associations. His professional interests are in the field of personalized diet, cancer, cancer-related fatigue, and homeopathy. Professor, you're welcome.
Congratulate all the founders and organizers of this important meeting. And hello to the panelists, and I just checked the participants. It's very heterogeneous, so I welcome them all as well. It is just that I really like the slides of Milena. They're really nice, but I mean, I have a small laptop, so I couldn't really look and see the details. So I hope to get the presentation when the event is over, finally.
Absolutely.
So my talk is on microbiome and nutrigenetics. It's a really general topic, and I'm just looking at the heterogeneity of the participants, so I'm just gonna make a sort of mixture, and I'm hoping at the end everybody gets a sort of message out of the presentation. So let's see. So I have no actual or potential conflict of interest in relation to this program or to my presentation. Those are my credentials, but you already have just mentioned them, so I'm just going directly into my presentation. We know that the microbiome has different effects on immunity and genetics, depending on the multiple factors, including location, and it's systemic versus local effects and interactions with the host. Not going into the details as much. In this slide, you see the mechanisms of microbial immunomodulation during tumor progression, because I'm mostly interested in microbiome and cancer treatment. Mucosal microbes can modulate the immune system locally or after translocating to the sites of the disease site. And microbes can either interact directly with the immune cells or provide indirect adjuvant cues for immune and gene modulation. And the consequent inflammation can be either pro-disease or anti-disease, with a diverse range of effects on genetics and epigenetics through the innate and adaptive immune system. Okay, so therapeutic microbiota may exert potential disease prevention through either enhancement of the gut barrier function or through immunomodulation with different, like dendritic cells, macrophages, even immune leukocyte antigens, etc. Promotion of an advantageous gut microenvironment that inhibits pathological bacterial colonization. We may have some effects through the selective cytotoxicity, but in this time the cytotoxicity is actually beneficial for the organism, and we also have the epigenetic and genetic modifications. Therapeutic strategies to modulate the gut microbiome can be through the microbiota selection or through dietary interventions. Microbiota selection, as you probably know, could be through probiotics or prebiotics or in combination. We also have the fecal matter transplantation and bacterial consortia. We can also play around with the dietary interventions, like we can have some plants, polyphenols, dietary fiber, or fermented food. And all goes to the ideal microbiota composition, right? I mean, to achieve the therapeutic effect. And nutritional genomics or nutrigenetics is an emerging area of research aiming to delineate the interplay between nutrients and nutrients intake and the reciprocal pathologies with the human genome, coupled with other omics, as you can see here, metagenomics, culturomics, metaproteomics, and others, along with nutrigenomics, aspires to individualized nutrition reminiscent of the pharmacogenomics and the individualization of the drug use. So we all know that the people are different in their requirements for and responses to nutrients and bioactive molecules in the diet. Many inputs contribute to the metabolic heterogeneity, including variations in the genetics, epigenetics, microbiome, lifestyle changes, diet intake, and eventually environmental exposure. Precision nutrition is not about developing a unique prescription for individuals, people, but rather about stratifying people into different subgroups of the population on the basis of biomarkers, and then using this stratification to better estimate the different subgroups of dietary requirements, and thereby enabling better dietary recommendations and interventions at the end of the day. So the hope is that we'll be able to subcategorize people into ever smaller groups that can be targeted in terms of recommendations. So I'm just going to give you a real quick example that diet-induced obesity and cancer and autoimmune-related protective effects can be achieved by a gut bacteria called Disosmobacter valbionis, which was first isolated in 2017. This bacterium is acting in an accompanied fashion by improved DNA methylation, histone modification, and non-coding RNA modification, affecting directly and indirectly autoimmune system adjustment, acid-alkaline equilibrium, and lower insulin resistance. And this is really very promising. And this is not only in obesity and diabetes mellitus. So we have some very preliminary data on cancer and autoimmune diseases. And we know that this valbionis is based on the data of the Human Microbiome Project or American Gut Project. And they found that this bacteria abundance was present up to 70% of the general population. So as such, determining predictive microbial biosignatures, which may be of clinical benefit through the GM sampling, the gut microbiota sampling, metagenomic sequencing, and bioinformatics has a major role to play in the future of gut microbial research. There are many other therapeutic strategies that can be played around, as you can summarize this in the slide. But with many studies are currently underway to elucidate the impact of nutrigenetics and microbiome. It's clear that this era of this science has just begun and improving exponentially. So not all epigenetic changes are permanent. Some epigenetic changes can be added or removed in response to changes in behavior and environment. I'm really very concerned about treating patients integratively. So you see the pyramid of healing here with different levels. Actually, we have 5 levels. There are some other people claiming there are more than 5 levels, but this is like a pyramid, and the first is always the physical body, the second one is the energy body, and third one is mental, continuing with intuitive and spiritual body. So when you do something, when playing around with the gut microbiome, you're mostly playing with the physical body. You're just giving it, either you're giving some nutrients or changing the gut microbiota content with fecal matter transplantation, etc. But we believe that, I mean, if you just don't treat or deal with the root cause, your treatment is not complete, right? So just kind of give you a very quick example that we are working on. For example, 85% of anorexia patients, they suffer a clinical zinc deficiency, right? So how do you treat anorexia patients in the clinic? You can just give zinc supplements, but you have to supply these zinc supplements lifelong. Let's see if I'm okay with timing. Okay, so, but if, I mean, you can just, while supplying zinc, you can just go to the second level and do some acupuncture, and this fecal matter transplantation also some effects on the energy level, which resolves the hidden malabsorption syndrome caused by overactivity of the sympathetic nervous system. That's simply actually precisely sympathetic celiac plexus, which vasoconstricts of the absorbing lymphatics and the blood vessels in the gut. We can also, while supplying zinc and doing the fecal matter transplantation or playing around with the nutrients, we can go through the mental level with psychotherapy or homeopathy or some other means, and we unresolve the memory held in the limbic system, which stimulates the hypothalamus, and at the end of the day, the sympathetic stress messages sent to the celiac ganglion again. So the third level has organizing effect on the second level here, and a vice continuing effect on the physical body, and vice versa. Without absorption on the first level, and a functioning autonomic nervous system in the second level, the patient wouldn't have the energy and functioning mind to work with it in a healing way. To summarize and finalize my words, I mean, I would say that diving deep in nutrigenetics and microbiome should not leave us blind, nor forget other disciplines and therapies, which certainly create shortcuts for achieving our goals. And for all the participants and panelists, whenever you happen to be in Istanbul, your table is reserved for your next personal and professional trip, and I appreciate your attention.
Thank you so much, Professor Diler. I will take you up on that because Istanbul is one of my all-time favorite cities.
Oh, thank you.
Okay, let's move on to our final speaker today. This is Dr. Nikolaus Gasche. He's a medical doctor by background, and he's gained experience at university hospitals through Austria, Germany, and Africa, which inspired him to numerous business projects. Nikolaus is the founder and managing director of Biome Diagnostics, a medtech company that is focused on transforming precision medicine through microbiome-based technologies and aims to advance precision cancer therapy, adding the world-class microbiome solutions to our clinical routine. Nick, you're free to speak.
Thank you very much. Can everybody hear me? Is my mic on? Great. And the presentation is visible. It's wonderful. Thank you very much, Veronica. Thank you very much, Biotech Atelier, and also NM Genomics for inviting me here today. Thank you also, Professor Diler, for the invitation and already introducing microbiome and cancer, because this will be one of my main topics now. I'm really thrilled to talk about my company, Biome Diagnostics, and how we actually want to bring the microbiome into the clinical area. But let me just quickly introduce us. My name is Nikolaus Gasche. I'm a medical doctor by background, as Veronica said. However, I've always been fascinated by technology, which led me to found a couple of companies in that area, of which I also sold one in 2017, and am now thrilled to combine medicine with technology together. Barbara, my co-founder, she did her PhD in Oxford and gained a lot of experience as a business manager at Siemens Healthineers, where she was responsible for multiple regions in Europe. We are a company of 16 employees now, are involved in multiple clinical studies with a large focus though on cancer, and have built up quite a large data set already. At the core, you can imagine us that we are a tech company. So we build upon next generation sequencing. So this is a technology that is actually used to identify the DNA of the bacteria. And what we do is the bioinformatics and the machine learning analysis afterwards to understand the gut bacteria and try to understand its impact on our health. And for that, we've also built up a knowledge database integrating thousands of publications to better get a feeling of the gut bacteria. What makes us special, though, is that this whole technical or the whole technology runs in an ISO-certified environment, which makes us the first company worldwide in the microbiome area to have an ISO 9001 and 13485 certification. We launched our first product in 2019 called MyBioma, which is a scientific do-it-at-home microbiome analysis where every European can actually get can take their gut health into their own hands. And for that, we provide a simple sampling kit. You would then take a stool sample, we analyze it and give you a scientific report on your personal microbiome in form of a mobile app or as well as a PDF report. Though the product is available online in Europe, we set a large focus also on white label partners and distributors where the analysis then maybe, or for example, can be provided under a different brand. And we can really concentrate on what we love to do and what we do best, the bioinformatic analysis behind it. And I just brought a fun fact there on the graphic below. With MyBioma, we aim to understand the European gut microbiome, and occasionally we do like retrospective analysis of thousands of data points. And in this case, we looked at, or this was a couple of days ago, we looked at the diversity of the European gut microbiome during the pandemic over the last 2 years. And just to get everyone up to the diversity, it's a simple calculation where you identify how many different species do you have in your gut microbiome and are they evened out. And the higher the diversity, you could say the healthier the ecosystem, whereas a lower diversity is mostly associated with specific conditions. And this preliminary graphic shows that there was a small dip in the diversity during the second and third quarter last year. This may be due to during the lockdown time, and that maybe we had it may correlate with different stress, with less human contact, maybe also with altered hygiene. It also may simply be coincidence. We haven't done any significance testing yet, but we will dive deeper into that the next weeks. And I just wanted to present something that came up a couple of days ago and that I thought was quite interesting. Well, we also see what Professor Diler was talking before is that the microbiome plays a crucial role in cancer care. And here we focus on two aspects. The first one is prevention, and the second one is therapy or treatment. So prevention includes early screening of, in our case, colorectal cancer, because we know that specific bacteria are associated with an increased risk of this cancer. And thus the microbiome would actually be an ideal biomarker to identify such a risk and to identify cancer in an earlier stage and to then treat it also earlier. And the second part is then the treatment, where we use the microbiome to improve immunotherapy outcome, and specifically immune checkpoint inhibitors. Immunotherapy is just maybe a general introduction on immunotherapy. It's a modern therapy in the oncology area where the therapeutic itself makes the cancer visible again to our immune system, and our immune system can then fight the cancer. You could say, simply said, the stronger the immune system, the better the cancer can be fought. But what is important to know is that the immune system that is largely located in the gut, and thus it seems obvious that the gut microbiome and the interaction with the gut wall can actually influence the training of our immune system. And thus we believe the gut microbiome is also a great biomarker to evaluate the strength of our immune system, and would then also allow to predict the effectiveness of immunotherapy, for example. And we're doing exactly that. So with Biome1, we are launching the first biomarker in the microbiome field. and it can predict the response and adverse events of immunotherapy, specifically immune checkpoint inhibitors. This test is non-invasive and works with a single stool sample before treatment initiation. What is also special is that Biome1 is tumor agnostic, meaning that it works across multiple cancer types. In our case, we're looking at 3 types. That's non-small cell lung cancer, melanoma, and renal cell carcinoma. As you can see on the top right image, in that case, we are correlating melanoma and non-small cell lung cancer, and we don't find any differences between responders and non-responders in those who have those 2 tumor types, which makes it actually a systematic biomarker. And below, we also presented that a list of bacteria where we found that they are specifically correlating with responders, and on the other side, we have bacteria that are not correlating with responders, but more with non-responders. So we are at the moment finishing up our clinical study with multiple university hospitals. We have 2/3 of the patient already enrolled, and we'll have a research use only device available soon. And I just wanted to give a sneak peek there. With Biome1, the report will include results on, for example, a benefit risk analysis, which is a combination of the response to, or the probability to respond to immunotherapy, and on the other side, the probability of adverse events, because that is still a main issue with checkpoint inhibitors. So the doctor would be aided in the decision-making process. Should maybe the patient be more closely monitored because of adverse events? Can maybe also more responders be identified for the treatment, for example? Lastly, I wanted to give a short outlook into the actually near future, which was also mentioned shortly, FMTs. So we believe that FMTs will also play a large role there. FMTs are simply fecal microbiota transplants, meaning that you take a donor stool and then transplant it to someone else to improve their gut microbiome health and to establish a healthy microbiome there. FMTs are nothing new. They've been around for many years. Some even say in the history they've been around since hundreds of years in different countries. And they're being performed in routine, for example, now with Clostridium difficile infection. However, there's emerging evidence that FMTs will be a game changer for immunotherapy, as, for example, this is one paper of many published this year that FMT can change immunotherapy refractory melanoma patients to become responders. But the biggest issue is still who would be the greatest donor or the good donor to make a patient then a immunotherapy responder. Who would be the equivalent of seronegative, for example, when looking at blood group, the blood group system. And this is something where we believe with Biome1 we can actually reinvent the blood group system, as we would then identify responders in the population at stool banks, for example, that can then be used as donors to transplant and improve the therapy outcome of cancer patients. This is nothing that is far away. This is, I can imagine this happening in the next couple of years. Yeah, if you're interested in our technology or you want to find out more about Biome1, please feel free to write me or call me, and I'm happy to discuss now at the panel. Thank you very much.
It was a great and interesting presentation. I am now opening the floor for a discussion. So we've received a few questions from our participants, but if you guys want to discuss something between you, I would prioritize this. So if you have, yes, Dr. Diler, Professor Diler.
Again, thank you very much. I mean, I really learned a lot from every panelist, and like your moderation as well. It's just that I mean, it's out of the scope of the discussion, but I see that there are some coupling points that I can do with the last speaker as well as Milena. So it's just that, I mean, when you're utilizing immunotherapy, it specifically, we have the impression that it specifically deteriorates the gut biota, but not all the lines, but in some lines. So it's a good way to see the progress and monitor the immunotherapy as well as the other therapies. I mean, which they have not yet used. So it will be good to connect and discuss further with the company. And thank you very much for everybody.
Thank you. We will send the list of participants, contact information, and so on after the 3-day event. So I'm sure you will have the chance to get to know more. Okay, our first question is for Dr. Georgieva. When you look at the health system as a whole, government, pharma providers, payers, doctors, and patients, what do you see as a driving force to promote and facilitate longevity? And when it comes to terms of data, what changes or support would you like to see to advance longevity medicine?
Well, thank you for this question. First, I also would like to say that the whole panel was really insightful for me, because microbiota, gut microbiota, and diversity in gut microbiota is very important for the aging process, because with the aging, the diversity decreases, and it is also a very good hallmark. So there are many clocks for aging based on the microbiota. And for the question is for $1 million because it tackles the whole problem from all parts. First, I see the first steps in translating the science to the public and also to the medical and pharma specialists. Then utilizing the efforts to get this information into research and development at these companies, because a lot of data are accumulated in the basic science and they should be translated to the practice as well. So I think all the efforts should be united, and this translation and symbiosis, as I already said, should be done at every levels. We scientists should communicate all the advancements. All the pharma and medical specialists should utilize this knowledge and start research and development with their patients and with their drugs, and then we can get to the market, because now we have the developments in this area very high, but the market is somewhere vague, somewhere around, and this transition should be made by the efforts of all of us. Because aging and is a whole ecosystem. It unites the immunotherapy, the cancer, also everything is united, and it's based also on the personalized diet, the personalized physical activity, the personalized therapies, the personalized gut microbiota treatment, and so on and so on. So we are together in the same battle, I think, tackling the health span, which has too much in common with the diseases that all we try to. So it's if I can understand and gather these specialists, I will do it. That's my mission, to translate this data that is at the basic science. I can discuss with everyone that wants to. Thank you.
Thank you for this answer. We have another question for Dr. Nikolaus. You said in your presentation that specific bacteria species are associated with cancer. Can you please explain which ones, and can these species be identified via the MyBioma tests?
Yeah, thank you very much. So what I meant with specific bacteria associated with cancer is, for example, with colorectal cancer, there's been a lot of research going on that even different stages of the cancers, let it be adenoma or already later stages of the cancer, like colorectal cancer, are associated with specific types. We've broken it down, so there are famous bacteria that act protectively or have a negative impact that have been published multiple times, like Faecalibacterium prausnitzi is a famous example, but we don't actually select like only 2-3 bacteria, but we have a list, or we've identified mostly correlations of 50 different bacteria that are relevant, and then build models upon that. We don't give a report in the MyBioma analysis on the cancer probability, because it is a scientific analysis that just shows what research is currently saying and how your specific microbiome associates with this research. But for such a cancer screening test, it would need to be CE marked. It would need to be approved regulatorily. So for example, with Biome1, which is used for immunotherapy, we are currently still conducting a study, but are already launching our performance study for the CE marking process, where the product is then evaluated, because you need to evaluate the accuracy. You need to know, is it more accurate maybe than existing methods, and what are the downsides? Yeah.
Thank you so much for this answer. I see that Professor Diler wants to say something.
Yeah. I mean, very good answer, Nikolaus. But it's just that it's not always. Referring the question. It's a great question, but we don't always get one bacteria to cause the cancer. It's multiple. But I mean, the work that I have mentioned very shortly, that there is this bacteria, Diagnostics valvionis. And the abundance of this bacteria in the healthy gut helps to prevent again. So it's not always causing cancer. So there are some bacteria protecting us to get cancer as well. So we need to monitor them as well as a preventive measure that I want to include.
I think that's actually really important to mention, that it's always also to look at the balance. So there are bacteria that can lead to such a disease or that are strongly correlated with it, but other ones that are actually also protective. And it's important to always look at the balance of those two groups.
Thank you, everyone. Unfortunately, we are out of time. So I hope that you will be able to pick this conversation some other time in another event. Thank you again to everyone. And I would like to remind our attendees that now we have a 30-minute lunch break, and we'll be back with our next panel.